So we are studying enzymes, right? And in the last video, you got to know about the definition, introduction and classification of enzymes. Today we will explore a bit more. Like we all know that there are two most striking properties of enzymes. And what are these? These are the catalytic property and specificity of enzyme, right? So we will start from a catalytic property, and while discussing this property, we will got to know about the specificity in more detail. So enzymes, which are basically globular proteins having a molecular weight not less than 100 kDa, I want to say that they are large proteins, right? But only a small portion of this large protein is actually responsible to catalyze a biochemical reaction. And this small site is known as the active site of enzymes, right? So active site is a region where the substrate binds along with cofactors, if any, and it also contains the residue which directly participates in formation and breaking of bonds, right? This is the enzyme and this is the substrate. Substrates bind to an enzyme, so binding takes place at the binding site, and there must be a formation of product, so there must be a catalysis, or there must be formation of new bonds followed by breaking of new bonds, and this all is done on the catalytic portion or catalytic site of the enzyme, right? So these residues which are involved in catalytic site where reaction occurs are known as catalytic groups. So enzymes which are classified on the basis of reaction they catalyze and the substrate on which they work on, they differ in the types of reaction they catalyze, in the types of substrate they work on, but there is some common feature which resides in the active site of enzymes and which remains common for all enzymes. And in this video, we will explore all these properties of the active site of the enzymes, right? Starting from the first one, that active site is a three-dimensional structure. Remember, in the protein lecture, I told you that although all proteins belong or consist of the primary structure, the open chain structure, but the original native conformation is the three-dimensional structure which is their functional form, correct? So active site is not a linear structure, it's a three-dimensional structure. Just keep this in your mind. So it is the site which is defined as a confined pocket on the enzyme, which is a region that binds to substrate, cofactors and sometimes prosthetic groups. The surface of the active site, which is a three-dimensional structure, it is lined up with the amino acid residues with substituent groups that binds the substrate and catalyze the chemical transformation. So I just want to say that enzymes, which are proteins, naturally they are made up of amino acids. So at the micro level, these are the amino acids, their groups, their side groups, which are actually involved in catalyzing the chemical reaction with the substrate. Okay. Since active site is three-dimensional in structure, residues from different parts of polypeptide chain can be present in the active site. If you are not getting this, let's take an example of lysozyme. This is the three-dimensional structure of lysozyme. Okay. So the X-ray diffraction analysis reveals that lysozyme has three subsites in the active site. which are located at number 35, 52, 62, 63 and 101. Okay. So the amino acids which are located very distantly, like after 35 it is 52, after 52 it is 62, and then 63, and then 101. So whether if active site is present in a linear form, is there any possibility of coming or joining or orientation of these numbers together? No. Since active site has three-dimensional in a structure, it is possible that the residues which are far apart in the primary sequence come up together in the closed environment when the protein or enzyme folds. Correct? So, since active site is three-dimensional in its structure, it is possible that residues from different parts of polypeptide chain can be present in the active site to perform the catalytic function. Clear? Coming to the second property, that active site takes up very small part of the total volume of enzyme. I already told you in the first slide that active site requires only a small portion of the total enzyme, and this small portion is as small as 5% of the total area of the enzyme, total surface area of the enzyme. So the question comes into mind that what does the extra amino acid do in making the whole enzyme? So the answer resides in that the extra amino acid serves as a scaffold or support to create three-dimensional structure. So three-dimensional structure is not possible by taking just 1, 2, 3 or 1, 2, 3, 4 amino acid. So there is a role of extra amino acid in creating the three-dimensional structure, and in many proteins the remaining amino acids also constitute regulatory sites, sites of interaction with other proteins, or channels which bring substrates to the active site. Okay, so the point to be noted here is that although it is the active site which is actually responsible for catalysis of chemical reaction, you can't ignore the extra amino acids for their supporting role. Coming to the third property, that active sites are clefts or surfaces. The active site in the enzyme molecules are grooves or surfaces from which water is largely excluded unless it is a reactant, and the favorable amino acids present in the active site are aspartic, glutamic, lysine, serine, etc. And the side chain groups like COOH, NH2, CH2OH, etc. serve as catalytic groups in the active site. So the active site which are clefts or surfaces, they are actually the grooves from which water is largely excluded unless it is a reactant. So you should remember this. Coming to the fourth property, that the charge distribution on the substrate and active site must be complementary. It means that all positive and negative charges must be cancelled out. Otherwise, there will be a repulsive force pushing them apart. For example, the non-polar character enhances binding of substrate as well as catalysis. So the active site usually contains non-polar amino acids, although sometimes polar amino acids also exist, and if they are present, they acquire special properties essential for binding or catalysis. So the active site contains certain amino acid residues containing certain charges, and the substrate contains some groups having certain charges. When they both come together, the resultant charge will be equal to 0, otherwise there will be a repulsive force, and which makes the enzyme destable or less stable. Correct? Coming to the fifth function, which is that the active site binds the substrate molecule by relatively weak forces. Whether we are talking about proteins or any other biomolecule, we found that these are actually weak forces which are actually responsible for stabilizing the protein structure, right? As discussed with the proteins, the weak molecular interactions which are hydrogen bonding and other electrostatic interactions, sometimes van der Waals interactions and hydrophobic interactions, these interactions hold the enzyme and substrate together in the complex. So covalent bonds sometimes exist, but the non-covalent or weak forces are the major forces through which the active site binds to the substrate and catalyze the chemical reaction. And out of these weak interactions, it is the hydrogen bonds which often provides high degree of specificity. By taking an example of enzyme ribonuclease, you can easily understand that the substrate of ribonuclease, which is a uracil, binds with the active site of the enzyme containing threonine side chain by hydrogen bonding. So enzyme ribonuclease holds the substrate through hydrogen bonding, which itself is a weak interaction. Correct? Now coming to the next property of the active site, and which is the final property of the most important property of active site, and which is the specificity. The specificity of binding depends on precisely defined arrangement of atoms in an active site. In the first slide I told you there are two striking properties of active site. First, the catalytic power, and the second, specificity. So specificity is there because enzyme and substrate interact by weak interactions, and a substrate must have a matching shape to fit into the enzyme. That's why a great scientist Fisher proposed a model for combination or coming up together to the enzyme and substrate, and this model is known as lock and key model. They were thinking that how enzyme and substrate come in close contact to give rise to the formation of product, and the first model or the first thinking which comes into their mind is the lock and key model. They suppose that the enzyme, active site and the substrate are complementary to each other, and just when substrate comes into contact with the active site, it fits into it to form the enzyme substrate complex, just like lock, which is the enzyme, and key, which is a substrate. There is one key per lock, as it is there is one substrate for enzyme. They come up and together form the enzyme substrate complex. So they concluded that the substrate must have a matching shape to fit into the site. Otherwise this reaction may not happen. Otherwise substrate cannot come into proper contact with the active site and formation of product cannot take place. So this was the first hypothesis which comes into mind. But we know that enzymes are not rigid, rather flexible in structure because they have three-dimensional structure. So another hypothesis comes into mind is that given by the scientist Koshland, and they believe that the substrate is not actually complementary to the active site initially, but when substrate comes close together to the active site modifies itself as per the structure of substrate, and then the proper enzyme substrate complex becomes fit into each other, or the substrate actually induces the conformational changes in the active site of the enzyme, which then together ease the formation of the enzyme substrate complex. And this provides an insight that active sites are complementary not to the substrates but to the transition states or ES, and this happens only after the substrate is bound. Correct? So we will discuss both these models one by one. Now let's explore a bit more the Fisher's lock and key model in more details. Like, this model is also known as template model, which is proposed by Emil Fisher in 1898. This is the another picture of the lock and key model of the enzyme substrate complex. So Fisher told that the union between the substrate and the enzyme takes place at the active site more or less in a manner in which a key fits into a lock. I explained in the previous slide, and results in the formation of an enzyme substrate complex, correct? And this is now clear to you. Now, only the correctly sized key, which is a substrate, fits into a keyhole, which is the active site, of the lock, which is the enzyme, correct? Now, this is portrayed as by assuming that an enzyme is conformationally rigid, which is actually not. So, the point to be noted right here is that the enzyme substrate complex is highly unstable, and almost immediately this complex decomposes to produce the end products of the reaction and to regenerate the free enzyme. Remember, in the previous lecture of enzymes, I told you that enzymes are not used up by the reaction. They do their work and becomes free. Just like the enzyme substrate complex is formed, and then when formation of product takes place, the enzyme remains free to catalyze the another reaction. Now the enzyme substrate reunion also results in release of free energy, and it is the energy which in fact raises the energy level of the substrate molecule, thus inducing the active state. And this is the very key point of the enzyme catalyzed reaction, that the transition states or the ES state is the highest energy state of any of the enzyme catalyzed reaction, and enzymes catalyze reaction by stabilizing the transition state. You need to be very, very clear to this point, that it is the transition state which is the highest energy state, and enzyme efficiently catalyze the enzyme catalyzed reaction by stabilizing the transition state, and this can be done by releasing of free energy, correct? And it is when activated stage, certain bonds of the substrate molecule becomes susceptible to cleavage and new bonds are formed, and which results in the formation of products, correct? Now coming to the second model, the induced fit model, as enzymes are not rigid in structure, they are having three-dimensional and flexible structure. So binding of a substrate often leads to a conformational change in the structure of the substrate or the substrates. This is the another picture of the induced fit model, in which you can easily understand that this is the structure of enzyme and this is the substrate. When substrate binds to enzyme, enzyme takes a conformational change in the active site and becomes fit into the and make it fit as per the structure of the substrate. You can very easily understand the induced fit model by taking an example of gloves. When you wear a glove, it becomes fit into your fingers, just like when substrate comes into contact with the active site of the enzyme, the active site made itself compatible or complementary to it to make it fit to the actual size or shape of the substrate correctly. This is the induced fit model. So this model you can easily understand that by taking an example of hexokinase right here on the top of the slide, that enzyme hexokinase, which has two domains, domain 1 and domain 2, and there are two conformations of the enzyme, open and closed one. Open, when its substrate glucose is not bind to the enzyme, but when substrate comes into the close contact to the hexokinase, it remains in closed conformation. It changes its conformation as per the structure of the glucose, so as the glucose moiety becomes fit into it, and now it is in the closed conformation. The hexokinase is now able to catalyze the chemical reaction. Correct? So after catalysis, the enzyme resumes its original structure as we discussed with the lock and key model. The structural changes that occur when an enzyme and substrate join together bring specific parts of a substrate molecule into alignment with specific parts of the enzyme's active site. Correct? Not all of the groups of the substrate does reaction with all of the groups of the active site. Only a specific part of substrate comes into alignment with a specific part of enzyme active site, which actually undergoes catalyzing reaction, or which actually undergoes in participation of reaction. So amino acid site chains in or near the binding site can then act as acid or base catalyst, providing binding sites for the transfer of functional groups from one substrate to another. Now it is very clear that why we discussed the lock and key model first and induced fit model secondly. The only difference is that the basic nature of the enzyme catalyzed reaction is same, like enzyme comes into the contact with substrate, ES complex form, and which results in the formation of products. The only difference is that the lock and key model assumes that enzyme has rigid conformation, while induced fit model, which is the more acceptable model, throws light upon that it is not rigid conformation, rather it is flexible and made it complementary to the substrate when substrate comes into the contact with enzyme, and there are few amino acid residues on the active site which actually takes part in the chemical reaction they catalyze. Okay, and this conformation change in the induced fit model takes place only when the substrate molecule joins with the enzyme. Correct? So this is all about the active site of enzyme, and in this video I want to make it very clear to you about all the properties of the active site which are common to all of the classification of the enzyme. You just need to make it very clear, because if your concepts are not clear, you will not be able to solve questions precisely, and the questions which I have selected for you today are all conceptual questions. Taking the first one, which of the statements regarding enzyme is false? And you need to read all the options very carefully. First, enzymes are proteins that function as catalysts. This is the true statement. Enzymes are specific. This is also true. Enzymes provide activation energy for reaction. Is it true? Does enzyme provide activation energy of reactions? No. So this is the statement which is false. You can read the rest two options also. Correct? The second one, the relationship between an enzyme and a reactant molecule can be best described as reactant means substrate. So the reaction between enzyme and substrate can be best described as, and the options are temporary association, an association is stabilized by covalent bond, one in which the enzyme is changed permanently, a permanent mutual alteration of structure, or non-complementary binding. And the correct answer is that temporary association. Enzyme always binds to the substrate mostly by the weak interaction, and these weak interactions are temporary because substrate has to release from the enzyme when the reaction is over. Correct? Coming to the last question, the active site of an enzyme, and the options are remains rigid and does not change shape, it's found at the center of globular proteins, it's complementary to the rest of the molecule, and contains amino acids without side chains. All these four options remain incorrect. So the correct answer would be none of the above. So you would be able to solve these kind of conceptual questions when you are clear with the active site and both the models which we discussed right here. And on the basis of this, I have also given you a homework question which is also a conceptual question, and you need to write a single letter answer in the comment section. And I'm sure you can do it now. Keep revising and happy learning.